Okay, so this section is about the tissue. Let's start with tissue. What is tissue? That's first. So tissue is a group of cells that have same function and similar structure. All cells, those group of cells that belong to this tissue must have similar structure and function. That's tissue. So first, then let's talk about types of tissue. Types of tissue. Now there are 4 types of tissue in general. 1. Epithelium. 2. Connective tissue. 3. Muscle tissue. 4. Nervous tissue. Okay. So that's 4 types of tissue. Again, tissue is a group of cells that have similar function and structure. Again, it cannot be 2 or 3 cells. They are not called tissue. It has to be a bunch of cells tightly attached together. That there is little difference between their structure or function. Okay, let's start with epithelium first. Epithelium. Now this is one type of tissue. So the question is, what is epithelium? Well, epithelium is any tissue that is primarily found on the inner and outer surface of bodies. What do I mean by that? It means that the inner lining of your stomach, intestine, heart, blood vessels, and respiratory system, lungs, airways, and so forth, urinary system, bladder, that's whatever the inside is covered by, it's called epithelium. Skin is an example of epithelium. Within the inner organs, the thin layer that coats the inside of organ is called epithelium. Okay? Alright. Now, epithelium is actually categorized based on 2 characteristics. How do we, for example, recognize different types of epithelium? So number 1, epithelium is categorized based on shape. Number 2, it is categorized based on arrangement. Okay, since we know that tissue is a bunch of cells that have same function and similar structure, so these bunch of cells could be recognized based on shape and how they're arranged in this case. So let's talk about epithelium in terms of shape. Now, there are generally speaking 3 types of cells possible. Okay, that makes up epithelium. So epithelium could be made of flat cells, okay, or it could be made of cuboidal cells or square cells, cuboidal or square cells, or they could be made of tall cells or columnar. So this is identifying epithelium based on shape. If epithelium is made of cells that are tall, then it will be called columnar epithelium. If epithelium is made of cells that are cuboidal or square, then it will be called cuboidal epithelium. If an epithelium is made of a group of cells that have the same shape, all of them are flat, then it will be called squamous epithelium. Generally speaking, epithelium is always identified based on the superficial layer. Whatever the superficial layer of that group of cell is, then it's called either cuboidal, columnar, and flat cells would be called squamous. So let me just use the appropriate term. Squamous. Okay, there you go. Now, next thing. So that's based on shape. Arrangement. Now, these bunch of cells, let's say they're all flat cells. These flat cells could be arranged in a single layer, or these cells could be arranged in multiple layers. All flat cells, let's say. So you can have 2 types of squamous epithelium. One that is only a single layer, and another one that's multiple layer. So that's based on arrangement. Single layer is called simple. If it's multiple layer, it's called stratified. Stratified. Okay, so similarly, now we know that you can arrange epithelium based on shape or based on arrangement of cells. If the cells are arranged in a single layer, it's called simple epithelium. If it's arranged in multiple layers, then it will be called stratified epithelium. Okay, let's do some examples to make it easier and simplify this. If we have bunch of cells, bunch of flat cells that are arranged in a single layer, then it will be called simple squamous epithelium. Simple squamous epithelium. So let's do this. Simple squamous epithelium. That's the one. If it's made of bunch of flat cells and they are arranged in multiple layers, as long as it's more than one layer, so 2, 3, 4, okay, then it will be called stratified. Stratified. But it's multiple layers, so that's stratified. It's all flat cells, so squamous. Squamous epithelium. Okay. How about if these cells were all cuboidal? Again, if it's a single layer, this is simple cuboidal epithelium. If it's multiple layer, more than one, it could be 2 or 4 or 6, doesn't matter, multiple layers, as long as it's not a single layer. Then it's called stratified cuboidal, so this would be stratified. Stratified cuboidal epithelium. Okay? So now we covered 3 types of epithelium, or actually 4. Cuboidal can exist in a simple form, single layer, or it can exist in multiple layer, stratified. Similarly, we have epithelium that is tall cells. Right? So let's say this is tall cells. All of these are tall cells. It's a single layer so far, so it will be called simple columnar epithelium. Simple because it's a single layer, columnar because it's tall cells. But as soon as you add more layers to this, more than one, now it is still tall cells, but it's not a single layer, it's multiple layers. So you'll call it stratified because it's multiple layer. Shape is still tall, so columnar. So this will be stratified. Stratified columnar epithelium. Okay, that's all for this. See, very simple, straightforward. Epithelium exists in 6 different shapes so far, or arrangement combined. So technically you can say there is 2 squamous type of epitheliums, there's 2 types of cuboidal epithelium, and there's 2 types of columnar epithelium. Okay, now there's an exception. One epithelium that's usually exception we should know, and then I'll give you an example as well of epithelium that you know exists which parts of the body. For example, now let's start with a unique type of epithelium. Let's see if you can recognize it. Now let's compare this. So let me erase this. So here you go. You tell me what type of epithelium is that. Usually we have to put nuclei in the center. You see, nuclei. Now you can see that all the nuclei are in a straight line, right? Therefore this epithelium has a single layer. And usually on microscope you'll be only able to see the nuclei. You might not be able to see the border of cells, so you focus on the nuclei. Okay, so single layer, so it's simple. Tall cells, columnar. Good, we're done. Let's do one more. Now, similarly, you have an epithelium made of tall cells, but it's multiple layer. Again, you might not be able to see their borders, which is okay. You will focus on the nuclei. Okay, so let's focus on nuclei. Here you go, blue nuclei. But again, you see there are 2 layers of nuclei. Okay, again, I'm going to draw a red line through it. One group, another group. That means this is made of 2 layers. 2 layers of tall cells. Okay, the term for multiple layer is stratified. 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 And the term for tall cell is columnar. Therefore, you will call this epithelium stratified columnar epithelium. That's what you will call it. And okay, good. Now again, I'm coming back to the unique epithelium. There are exceptions sometimes. So let's do the exception. Let's see if you can identify this one. Okay. Dark nuclei. You see, if you pay attention to this, again, you might not be able to see under a microscope the border of these cells. And if you look at the nuclei, they're not lined in a straight line, not in a straight horizontal line. They're all over the place. Up, down, you know, they're spread randomly because now if you think about the cells, you might say this is tall cells, so let's say columnar, which is correct. These are tall cells, so it's columnar epithelium. But if you look at it, is it a single layer or multiple layer? It appears as if it's multiple layers, but it's actually a single layer. So it's still simple columnar epithelium, but really you can't determine that just by looking at it, because all the cells are attached to the bottom base membrane. But because they're twisted, like as if you twist your fingers, it appears as if it's multiple layer. So it is not truly stratified. It's fake, pseudo-stratified. Pseudo-stratified, stratified epithelium. Again, there you go. Also, in some cases, epithelium might have on the surface cilia. And then you will call it pseudo-stratified ciliated columnar epithelium. So you add the term cilia to it. Okay. So far we have identified, let me write down these names. We have identified epitheliums. Okay. We have simple, squamous epithelium. Okay. So flat cells can be arranged in a single layer or multiple layer. Now, an example of this would be, for example, alveoli. You will find this in alveoli of your lungs. And you will also find it on the inside of blood vessels. There it's called endothelium. So I'll just say vessels. All vessels, blood vessels or lymphatic, they will have it. So you'll find simple squamous epithelium where? Within blood vessels and within alveoli. Okay. Let's say now you have stratified squamous epithelium. Okay, where would you find that? Skin is a good example. Esophagus, a great example. Okay, and of course birth canal or vagina. These are some locations where you'll find stratified squamous epithelium. Now you might have trouble memorizing that, so what are you going to do? Let's make sense out of it. If you have a single layer of cells, flat cells, right, compared to multiple layers, now which one would have easier time? Which one will be easier for transfusion or diffusion? If you have single layers, that means molecule can go in and out much easier compared to epithelium that is made of multiple layers. So stratified squamous, flat cells, right? That means this is made of many layers, many flat cells. Okay, so I would think it's easier for passage of molecule through a single cells compared to multiple layers. So this is why, since alveoli of lungs are used primarily for diffusion or transport of oxygen and carbon dioxide, it must be single layer. Common sense. Blood vessels, for example, capillary, again, it's a site of exchange for waste in nutrition, oxygen, carbon dioxide. So exchange, transport across it. So it should be made of single cells or a single layer. When you compare that to stratified squamous epithelium, think of skin. Skin, you don't want things, for example, to lose water every day. So skin must be made of multiple layers of flat cells for protection. Esophagus needs protection. It needs to be made of multiple layers because you eat sometimes crazy food. Some foods are harsh, some are rough, some are smooth. So the rough food can cause erosion, friction, and so forth. You know, birth canal, vagina, again, you need multiple layers because of intercourse can cause damage to it. Skin, again, you come in contact with surfaces, bugs and stuff, so it has to be made of multiple layers. So usually, in general, stratified squamous epithelium is primarily for protection. Okay, and simple single layer of squamous epithelium is primarily for diffusion. It allows diffusion, it permits diffusion. So, okay, so hopefully at least you know these two for now, and you should memorize them. Where would you find these? Okay, now cuboidal can be stratified or simple. Okay, usually stratified cuboidal epithelium don't exist that much in the body, so it's not important. Simple cuboidal epithelium, you find them in the thyroid, follicles, okay, and maybe renal tubules or nephron. So that's cuboidal. Now columnar epithelium is more important because you'll find it in many locations. So columnar, we can have three types. Let's say this is columnar. Columnar cells can be arranged in a single layer. So it could be simple columnar. It could be arranged in multiple layers. Let's say in this case it will be stratified. Or there's an example of them that's pseudostratified. It's not truly stratified. It appears to your eye as if it is multiple layer. So again, columnar epithelium can exist as a simple columnar, stratified columnar, or pseudo-stratified columnar epithelium. Good. Simple columnar epithelium is found in the GI tract. Not esophagus, which is part of GI tract. So we're starting from stomach to anal canal. You'll find it there, and it makes sense. Now, why would GI have simple columnar epithelium? Well, the primary purpose of GI is secretion and absorption. You need tall cells that can secrete, for example, secrete substances like mucus, right? Enzymes for digestion. Okay. Also, it should be able to absorb. So it needs to be bigger cells so it can store more, again, for absorption. For example, you want to absorb amino acids. We have discussed this before. Or glucose, or monosaccharide, or lipids, or electrolytes, like sodium, electrolyte. Okay, so that's the purpose of tall cells. Tall cells have much more room. They're bigger. They're much bigger compared to flat cells or cuboidal cells. Therefore, it makes sense that the GI has to have large cells which can store more and release more. Okay, so and it has to be a single layer. So because multiple layer, again, for transport, secretion, absorption, it's difficult. So single layer is important. Okay, stratified columnar epithelium. You primarily, for example, you'll find this in the uterus. In the uterus, you know, you have to have multiple layers so the fetus, the baby, can have much more nutrition. Pseudostratified is important. Now, the pseudostratified epithelium is usually found in airways. What do we mean by airways? Trachea, bronchi, and bronchioles. Okay? It's a single layer, but it's twisted around, so it appears as if it is stratified, but it's truly not stratified. Okay? And here it's ciliated as well. So you'll call it ciliated pseudostratified epithelium. Good. So these are some epithelium, or in their example. And location of the body where you'll find them. Let me go to one more thing. Now for epithelium, all epithelium will have surfaces. All epithelial cells. Let's say this is one cell, and this is another cell. Remember, we talked about that epithelium is a group of cells that have the same function and structure. Epithelium can be made of cuboidal cell, columnar cells, or flat cells. But let's talk about columnar cells. Okay, let's talk about columnar cell. Bad diagram. So I'm just going to draw 3 of them. Hopefully we can make sense out of it. Okay. So these columnar cells, let's say in this case 3 columnar cells. 3 columnar cells. Now that's a bunch of cells that have the same structure and same function, so we'll call them tissue. In this case it's epithelial tissue because it's found in the GI, let's say, or kidneys. Now they should have any box structure, like a box structure, should have 6 surfaces. Generally speaking, epithelium must have in the bottom a basement membrane. Okay, so all epithelium are attached to a basement membrane. Okay, let me just do this. There you go. Red is the basement membrane. So all epithelium are attached to basement membranes. It's for the strength and it's for stability. Now let's identify some areas on epithelium. Some surfaces. This surface, yellow, I'm coloring yellow. This is known as the apical surface. Apical means top, not the bottom surface. So epithelium at least has, the cells of epithelium will have apical surface. Only one surface of the epithelium should be apical surface. Okay, good. Now epithelium in red, right here, has basal surface or bottom surface. This surface faces the basement membrane, therefore it's called basal surface, and it also points towards the blood vessel. So let's talk about dark blue. Now you have surface on this side. These are called the lateral surfaces. Lateral surfaces. Lateral surfaces. Okay, so that's epithelium. Epithelium should have these surfaces. Okay. The basal surface is attached to basement membrane by hemidesmosomes or integral proteins for strength. Okay. Now the lateral surfaces are tightly attached by desmosomes, by actins, or in this case let's call them tight junctions. Tight junctions right here. These are tight junctions. The purpose of these tight junctions is to make sure that molecule cannot diffuse across. So therefore these tight junctions will create barriers. Number one, it holds these cells together as a group for stability. Second is to create these barriers. Tight junctions will create barriers. So that means things cannot go on the opposite side. Okay. On the apical surface, sometimes they will have cilia or villi or microvilli, depending if it's respiratory or GI epithelium. So please memorize these surfaces. Epithelium should have 3 identifiable surfaces, not anatomically but functionally. One is apical, which is yellow, apical surface or luminal surface, blue, lateral surface, and this red right here is basal surface. Now all epithelium will have one basal surface. All epithelial cells will have one apical surface, but it will have 4 lateral surfaces. 4. If you think of a box should have the top and bottom. The top is apical surface, the bottom is basal surface, and the sides are lateral surfaces, 4 of them. Good? Note that epithelium are attached to, these cells of epithelium are always attached to each other tightly. through tight junctions, okay, which prevents diffusion and it creates concentration barriers. Okay, good. So that's epithelium. Now let's go to another type of tissue. The next one is connective tissue. And the last lecture, Friday's lecture, we only talked about epithelium. So two epithelium, connective tissue and epithelial tissue. Muscle tissue and nervous tissue we will not cover now because that's part of the nervous system or muscle part. Okay, let's move on to connective tissue. The second type of tissue is known as connective tissue. Connective tissue. Okay, connective tissue. How do we define that? Any tissue that is not a tissue that is not muscle, that is not nervous tissue, and that is not epithelium. Remember we talked about earlier, we talked about it that there are total 4 types of tissue. So connective tissue is everything as long as it's not muscle, as long as it's not nervous tissue, as long as it's not epithelium. Everything else is connective tissue. That's the best way to define it. So that means there's too many types, and we're not going to spend a lot of time because different tissue, different system of the body will cover it. Okay. Connective tissue. Now we need to talk about cells of connective tissue. Let's talk about cells of connective tissue. Okay. Some of them, not all of them. Of course, is white blood cell nervous system? No. Are they muscle cells? No. Are they epithelium? No. So white blood cells will come under the category of connective tissue. Okay. Anything else? Adipocytes. Adipocytes, which means fat cells. Are they nerve cells? No, they're not. Are they muscle cells? No, they're not. Are they epithelial cells? No, they're not. So now, by default, they have to be connective tissue. Okay. Let's talk about fibroblast. Fibroblast. Again, they're not muscle cells, they're not nervous system cells, and they're not epithelial cells. So they have to be what? Connective tissue. Okay? And I want to talk about these ones and we're okay. All I want to do is identify their structure and we have to know their function. And that's good enough because we will discuss them further in the appropriate system. For example, we'll talk about Wbcs and the immune system, right? Okay. So again, we're talking about connective tissue. Let's talk about white blood cells. Okay. White blood cells. Again, this is not an immune system lecture. Here you just want to know the shape and function of these white blood cells. Generally speaking, there are 3 categories of white blood cells. 3 categories. Lymphocyte, lymphocytes, monocytes, and granulocyte. You should memorize that and you'll be okay. Let's go first to lymphocyte. There are many different types of lymphocyte. Let's talk about them. At least 3 of them. Okay, so T cells, B cells, and natural killer cells. Okay. T cells have further 2 types. T cytotoxic cells and T helper cells. B cells can become plasma cells. I'll put P cells. Natural killer cells will stay natural. Monocytes will become macrophages. Macrophages. Okay. But again, macrophages in different tissue would have specific name. In bones, they're known as osteoclast. Osteoclast. In nervous system, they're known as microglial cells. Okay. And in liver, they're known as Kupffer cells. Okay. In skin, for example, known as Langerhans cells. Let's go to granulocytes. Granulocytes also have three types. Okay. All of them will end with phil. All granulocytes. So we have eosinophil. We have basophil. And we have neutrophil. So all of them should have the term phil in them. And here you go. All of them should have that. Okay, all three of them. Okay. That's white blood cells. So let's go now to lymphocyte. How do we identify them? Again, so you know them based on names at least. Let's name them. Let's get to know them based on function, and then I'll tell you their structure. Okay. Neutrophils just need to know one word: kill extracellular bacteria. And generally, they kill bacteria. They can kill any bacteria, but mostly they deal with extracellular bacteria. Basophil release histamine. They produce histamine. So for that reason, histamine is the cause of allergies, right? So they will cause allergies or allergic reaction for that matter. Eosinophils, they kill parasite. Kill parasite. How they do it, it's not important right now. We'll do that in the immune system. Macrophages. Macrophages can actually neutralize, kill almost anything. They don't have any specificity. They're not prejudiced in that matter. Okay, so, but something unique. Again, macrophages will engulf, swallow, phagocytose, or kill anything. But let's talk about something specific that's unique to macrophages. They kill fungus. Okay, they also kill intracellular or atypical bacteria. Okay, and we have to memorize their name, bacteria. That's something unique, but other than that, they can do anything. They will even, for example, swallow smoke particles. Okay, P cells or plasma cells in B cells, they produce antibodies. I'll put AB. Here you go, antibodies. Okay. Natural killer cells will kill any cell that does not have their driver license or the ID card. And the ID cards are known as MHC1. So any cell that lack MHC1 will be killed by who? Natural killer cells. Okay? So keep that simple. T helper cells don't kill anything. They simply make phone calls. They rearrange the immune system. And they organize attack. Cytotoxic T cells will kill any cell that carries, kill any cell that carry abnormal ID. For example, in the United States, if somebody has Chinese passport, Afghan passport, or Mexican passport, and they're here illegally, maybe the immigration will catch them and say, what are you doing here? You don't belong here, in that sense. So abnormal ID. And in this category, the abnormal ID is MHC1, abnormal MHC1. Okay, so in general, there are three types of cells that are killed by cytotoxic T cells. Tumor cells, okay, transplant cells, and viral infected cell. Three types of cells killed by cytotoxic T cells. Good. So that's their function. Let's go now into what do they look like. So let's go to their structure. Okay. We'll go to their structure. We talked about their function, what they do, and now let's talk about them, what they look like. Let me just erase all of this and go here. Okay. Let's go to monocytes. Monocyte. What do monocytes look like? Usually they will have irregular shape, a single nucleus, maybe some granules. Okay. So they will have a single nucleus, that's monocytes. Okay, and then they will have some granules. I shouldn't use blue, let's use a different color, gray. Okay. again, they don't stain any different. They all stain almost like see-through dark blue, the nuclei. Okay, that's monocytes. Let's see how they are different than lymphocyte. All lymphocytes look the same. All lymphocytes look the same. Okay, so it's still recording. That's good. Lymphocytes. All lymphocyte will have a single nucleus. All lymphocytes should have a single nucleus that usually stains dark blue, and then they will have a thin cytoplasm. Look the ratio of the nucleus compared to cytoplasm. They have a huge nucleus. They don't look like monocyte. You can clearly identify them. Monocyte have a single nucleus that's small, but they have granules. Lymphocyte don't have granules, but they have a large nucleus and clear, small, tiny cytoplasm. Let's go on to granulocytes. Granulocytes, there are 3 types of granulocytes. Okay, 3 types of granulocytes. Again, number 1, they're called granulocytes because they have granules inside their cytoplasm. Okay, they have granules inside their cytoplasm. Okay, good. So let's add granules. Now the cells that stain, the granule stains blue, you see these dots? They're like granules, little small vesicles, bags. Any granulocyte who's, so they have granules, all of them should have granules, right? Some have red granules, some have blue. Here you go. This one has granules too. And let's talk about this one. This one has granules, which don't stain. Okay, so how do you identify them? All of them must have granules. That's one thing, but all 3 of them should have more than 1 nucleus. So some have 3 nuclei, some have 2, but never 1. Never a single nucleus. Okay, so never a single nucleus. So there you go, that's the nucleus. This one has 2 nuclei. This one has 2 nuclei. Okay. The one that stains red is known as eosinophil. Eosinophil. Again, all granules have granules and multiple nuclei. That's how you identify them. The one staining red is eosinophil, or its granule staining red is eosinophil. The one that's staining blue, blue B, basophil. The one that does not stain, it stays clear, and that will be called neutrophil. Okay, neutrophil. Neutrophil is neutral. Neutrophil. Neutral, so they don't stain. Okay, that's how you should be able to identify these cells, and these are white blood cells. You know their function and you know their structure. Okay, adipocytes usually will look like a vacuole, clear vacuole. Let's say this is a cell. Okay, in this cells, they will have a small nucleus, usually because they store a small nucleus. Okay, and then they will have a huge cytoplasm. In adipocytes, what do? They store lipids. And the type of lipid they store is known as triglyceride. Triglyceride. So adipocytes are nothing more than a storage for fat or lipid. Okay. In order for them to store, they store glucose too, but it has to be converted to lipids. So that's adipocytes. Okay, what else? Fibroblasts. Fibroblasts, these cells, you know, they have huge, for example, large nucleus and nuclei, and very developed endoplasmic reticulum, especially rough and Golgi. Why? Because they secrete collagen. Collagen is a protein, and this collagen is a linear protein, like a rope. It looks like this, so a wavy form of protein. Let's say there's a tree, like tree hair or braided hair. That's what it will look like. That's collagen. So if it makes protein, the nucleus has to be large. Endoplasmic reticulum has to be large. I'll put N for nucleus. Rough ER, let's say rough ER, has to be large because that's the function of rough ER in the nucleus, to make this protein. So they should have a lot, huge nucleolus and nuclei and rough endoplasmic reticulum because they continuously produce collagen. Okay, and these cells are found in the extracellular spaces, not within other cells, and their products. So their purpose is to give you strength and so forth and protection. And okay, let's take a break.